Welcome everyone to today's talk by Soledad Villar. I announce her as researcher from NYU, but this is not actually that correct anymore. Semi-correct. Now from Johns Hopkins. I know her as an expert in clustering, but recently she's also been working a lot on deep learning. Today, I think this is a talk more in the direction of obviously dimensionality reduction, what the title says. We're all very excited for this talk, but before I can hand over to you, I just want to say something about the procedure of this seminar. So please feel free to ask questions during using the chat. I will monitor this chat, then occasionally maybe interrupt the talk. But there's also a co-author of Soledad who can also answer at some point. So please do that. And now please go ahead.
Well, thank you so much for inviting me to give this talk. This is the first talk that I gave as affiliated to Johns Hopkins. And I see that a lot of my colleagues are here in the audience. So thank you so much for supporting me. I'm going to talk about dimensionality reduction and matching datasets. Some of these I already talked about when I went to Johns Hopkins last time, but I also have some new results that I'm going to incorporate to this talk. So basically, the idea is to discuss two fundamental data science problems: dimensionality reduction and matching data sets, like finding correspondences between data sets. And the motivation of this talk is processing single-cell RNA sequencing data. So, but I'm not an expert on the computational biology side. I work more in the math and the algorithmic side. I have my collaborators in the computational biology side that maybe are in the audience, and maybe Bianca can help me answer questions in this, if questions in this area show up. So basically, there exists this new, relatively new technology that allows to compute the genetic expression for individual cells. So basically, say in this example that I have here from the NYU Satiha lab, we have this data set of blood cells where we have 8,000 cells. And for each of these 8,000 cells, they can compute the genetic expression, meaning how many times each gene is expressed in each cell, of 36,000 genes. So this, you can think of this as 8,000 points in dimension 36,000. So what do you want to do? You want to identify genetic markers that satisfy a certain purpose. So that's the genetic marker selection. Just choose some genes that would give you relevant information about your data set. And the other problem that I'm going to discuss is the problem of matching. So say that you have a similar set of cells, and you can produce the gene expression with a different experiment, so with a different machine. And then there's some bias that is introduced by the technology. So you want to find a way to align these data sets, find correspondences between these data sets, and remove these, what is called batch effects. Or maybe you have data from different modalities. So you may have data from single cell expression, but also you can have chromatin accessibility data, and you want to find the correspondence between these two data sets. So what cell expression corresponds to their chromatin accessibility. And so this is a hard problem, the matching problem. We're going to discuss both of them and the idea of how to combine ideas from marker selection and matching in an unsupervised way. So let's start with the marker selection problem. Say that we have this data set of single cell RNA sequencing, and it looks like this huge matrix where a lot of them are zeros. It's like, say, it's very sparse. 90% of the entries are zero. And you want to visualize this data set. So one thing that people do for visualizing this data set is using TSNE, which is basically it's like a visualization technique that has many issues, but is widely used in this field. What they do is they consider a probability distribution between pairs of cells in this space, and they consider a probability distribution in dimension 2, and they want to do is they want to find a map from the high dimension space to the low dimensional probability space that minimizes the KL divergence, and they do this using the stochastic gradient descent. So that is one way of visualizing this that has many issues. Other ways to visualize this is like this algorithm called UMAP that is based on manifold learning. Another, after you visualize this data set, you may want to cluster it. There are many algorithms that you can use for clustering. For instance, k-means or k-nearest neighbors. In this example, I'm not. Of course, you will never do the clustering on this two-dimensional space, but you would do it in the high-dimensional space. And in this case, I actually don't need to run clustering in this data set because I know the labels of this data because the data is annotated. So this is like a supervised problem where I know that for each data set, for each cell, I know what kind of cell it is. Like I have different cell types, and there is and this data set is annotated. The way these data sets are annotated is a very complicated problem that I'm not gonna get into, but let's make the assumption that we do know the classification structure of the data. The marker selection problem here asks for find a very small number of genes, so that when you restrict this data to these genes, then you maintain the same classification structure that is given to you with the labels of your data set. So basically, one way to visualize this is like, if I do this projection of my of this TSNE visualization of my data set in 36,000 dimensions, then I get this classification structure. And when I just do the same thing, but now my 8,000 points are restricted to these 20 dimensions of the markers that I choose, the classification structure is maintained. Even though this looks very different, you can say that, okay, it's maintained in a way. So the reason why this is an important problem in computational biology, well, there are many reasons, but one of them is the fact that the sequencing of these cells is a biochemical procedure that destroys the cell. So, and also is very expensive. But there are other imaging techniques that they have, where they can have an electron microscope, where they can select some genetic markers and say that one may want to light up the cells that have some high concentration of certain genes. So here, the high concentration of this gene is marked with red. And so one can combine, like one can select what markers to use using this genetic marker selection that is highly correlated with the clustering structure, and understand, say, the position of the cells with that marker within the tissue. So you can use both. Like you can use both technologies in a complementary way. So, so basically this is a dimensionality reduction problem from dimension 36,000 to dimension 20. There are many techniques in data science that one can use for dimensionality reduction. I'm not gonna describe them all, but what I just wanted to say is that different dimensionality reductions techniques are most effective for different problems with different objectives. So for instance, these random projections that had the property, like if you can use a random projection when you have very few points, you can go to dimension order of the log of the number of points and preserve the pairwise distance, for instance. If you want to preserve the pairwise distance and you have few points, then this would be a very good way to go. You can also use autoencoders where you start with a high dimensional point data set, you reduce it to a lower dimensional latent space, and then you reconstruct the original data set from the lower dimensional latent space with, and you can learn this representation using deep learning, etc. So there are many, many ways to do dimensionality reduction. And in this talk, I'm gonna talk about this SqueezeFit dimensionality reduction technique that was introduced by us, like Dustin and Colbert McWhirter, and Dustin is in the audience, and I. And the idea is, okay, this is a dimensionality reduction technique that is based on preserving the classification structure of your data, that this is what we want to do here. So a more general mathematical formulation of the problem is this called projection factor recovery, where say that you have data in a high dimensional space and you're looking, and this data is labeled. So this is a supervised problem. And basically what you do is you want to find an orthogonal projection onto a subspace so that when you project your data, you project the data and you keep the labels, and the classification problem becomes easy or easier after projection. So I have an example that I can show you here. So say this is a toy data that I produced in MATLAB. So we have this data in dimension 3, and we have two classes, the red crosses and the circles and the green circles, and we want to find a projection into a lower dimension space so that the classification is easy. So if we do PCA, what happens is, like, since we have such a large component in the Z direction, then the PCA doesn't capture a direction where these two data sets are easy to separate. Even if you consider the different set between red dots and green dots, and you do PCA on those differences, since you still have a very high, large variation in the Z-axis, you don't get to separate these things properly. The same thing happens with LDA, this linear discriminant analysis, because LDA, what it does is, like, it assumes that these two classes are Gaussians and then finds a hyperplane to separate two Gaussians. And here, these two data sets, we're gonna see that are not linearly separable, so this won't work very well. So what we're going to do is we're gonna do SqueezeFit, that this is the algorithm that we're going to introduce that actually identifies that in order to separate these two sets properly, the direction that you want to project to is the XY plane. And I know that this is the XY plane because this is the way I constructed the data. Like, I constructed two concentric circles, and then I added noise in the Z-axis. So this is the projection factor recovery. Identify a subspace where the classification, when you project to a subspace, the classification is easy. And I think this is the most important slide of the talk. So maybe bear with me. So what are we going to do? How are we going to formulate SqueezeFit? Basically, we're going to do this. Say we have these two classes, class 1 and class 2. I'm going to consider Z a set of differences between points in class 1 and points in class 2. So all these Z vectors. And then what we're going to do is we're going to look for a projection so that to a lower dimensional subspace, so that when I project to that subspace, all these Z vectors get projected to something with, to some vectors with lengths at least delta, right? So this is an idea of kind of like what support vector machines does, like find a margin between sets. But here, the margin is not going to be like a linear separation between the two sets, but just say that 2 points from different sets need to be projected to something with distance greater than delta. So the way we write this is we're minimizing the rank of a projection so that when we project these difference points, the length is greater or equal than delta. And we restrict ourselves to orthogonal projections. That's the formulation of the problem that we want to solve. Optimizing in the space of orthogonal projections is hard because this is not a manifold and it's not convex. So what we do is we're going to relax this set of orthogonal projections to a convex set where optimization is tractable. So what we do is, and maybe this is also, this is a slide where I explain what the semi-definite relaxation does. So the idea is the following. So these orthogonal projections are matrices that satisfy that pi is equal to pi squared because it's a projection. The singular values of this matrix are either 0 or 1. And so what we're going to do is we're going to relax this pi square to a matrix M, which instead of asking that the singular values are either 0 or 1, we're going to say that this is a positive semi-definite matrix that is smaller than the identity. So meaning that the singular values are between 0 and 1, in the interval 0, 1. So that's the first relaxation. And then the rank of a projection is just the number of singular values that are non-zero. So it's also, in this case, the projection, so it's also the number of the singular values that are equal to 1, and it's just the sum of the singular values. So if we go to this space, it would be translated into the trace of this matrix M. And so so now we are going to say that this M is a relaxation of this pi square, or say the square root of M is going to be a relaxation of pi. So the constraint of asking that the length of pi z is greater or equal than delta gets translated to this quadratic form on z is greater or equal than delta squared for all z in my difference set. So the semi-definite relaxation of this optimization program is to minimize the trace of a matrix M subject to this, for all z in my difference set, z transpose mc is greater or equal than delta squared, and M is positive semi-definite and greater than, and smaller than the identity. In order to use SqueezeFit for dimensionality reduction, what we do is we solve the semi-definite program, and then we take the square root of the optimal M, and that's going to be a proxy for our projection. If the trace of M is like the L1 norm of the singular values vector of M, or and so you can think of this L1 norm, minimizing the L1 norm promotes sparsity. So you can think that a lot of zeros are going to show up. So you expect that this phi is going to be low rank. And so it's not going to be a projection in general, but you can think of it as a proxy of a projection and train your favorite classifier on these projected x's and keep the same label. So we can theoretically analyze this semi-definite relaxation in the following way. So first, we have this semidefinite relaxation, and we're going to say that a set of vectors is delta fixed if you take the solution of the semidefinite program and you take the square root of that and you apply to your x's, and the x's don't move. So here in this example, if I have that my delta is the distance between a point here and a point here, these are concentric circles, so the distances are the same. There's no projection that can make them larger, or there's no way you can move these two sets with a projection. So the solution is going to be the identity restricted to the points. And we're going to call contact vectors the shortest members of the difference set if they exist. So here, say we have these two data sets, and the shortest difference between x's and zeros are these ones, say. And those are my shortest members, say that they all have the same length. So we can say that, we can prove that, well, it's easy to see that a set is fixed by SqueezeFit if and only if the solution of SqueezeFit is the orthogonal projection onto the span of the data. And you can show that once you apply SqueezeFit, then the data is fixed. So you cannot squeeze this many times with the same parameter delta. And for finite data, your data is delta fixed if and only if the contact vectors span the entire data and have length delta. So this is not true for infinite data, because, for instance, here these two data sets cannot be squeezed anymore if you use this length delta as the parameter in your semi-definite program, but you don't have a basis of contact vectors because this is an infinite set, and so it gets more complicated. But for finite data, this is true. So what we can prove for SqueezeFit is this projection factor recovery problem that we discussed at the beginning, that claims that if your data is constructed so that you plant like is constructed in the same way, kind of like I explained, concentric circles and then noise in the z-axis. So if you know that there exists subspace T, so that when you project to this subspace, your labels are a function of the projected data, then you can recover this planted subspace, provided that the data in the planted subspace is well behaved, and the data in the orthogonal complement is independent of the data in T. So this is just like a meta theorem, and we have a couple of instances of it saying like what we can actually prove in this setting. The simplest example is the following of this theorem. Say that you have A, that is just some points with some labels, and what you do is you take some subset included in the orthogonal complement of the span of the data, and you add that to every point. So you consider B, which is every point in A gets added every point in S, and with the same labels. So if you do that, then like M is a solution of a SqueezeFit for A if and only if it's a solution of SqueezeFit for B. This is like a very simple example, because if you just look at the set of constraints, how they look like, you always add S to every point. So the set of constraints are actually a subset like the set of constraints in A are a subset of the set of constraints in B, and that's easy to analyze. So in this case, this is a very simple example where we don't assume anything for A, but we assume a lot of structure for what you're doing in the orthogonal complement. So maybe this is not such an interesting example. A more interesting example happens where you consider A to be a data set that is delta fixed, and then you add noise, Gaussian noise in the orthogonal complement, something similar to what we did in the concentric circles example. And the Gaussian noise is supported in the orthogonal complement of the data. Then if you have enough samples and your signal to noise is large enough, then you can show that the solution for for B is the same as the solution for the original problem. So adding the noise doesn't change the solution. The notion for the SNR has to do with how well conditioned are these contact vectors that we discussed before, like how well conditioned are these difference vectors that have the shortest distance. So here you can think that if these are the shortest vectors of length delta, they are actually linearly independent, but they're almost parallel. So the condition number is very large. But in this case, they are better conditioned, so the SNR is higher. That's the intuition. And the way we prove this is using dual certificates. So basically, if you have a convex problem or a semi-definite program, you can write its dual problem, and that's like a standard thing. There's this weak duality property that tells you that the value of the semi-definite program is always greater or equal than the value of the dual. In this case, the semi-definite program is a minimization. And the strong duality property tells you that for the optimal, the value of the primal is equal to the value of the dual. So given, if you have a candidate solution for your primal problem, you can construct a dual feasible solution that has the same objective value, and then that's a proof of optimality. So the strategy for this problem is to start with a dual certificate for the set where you know what the solution is for A, the set without noise, and then construct a dual certificate for the set with noise. And in order to do that, we use two ingredients. One is concentration of the spectrum of random matrices, and the other one is we need to show that there exists a point satisfying certain constraints, meaning that the intersection of a cone and a linear space is non-trivial. And in order to do that, we use the kinematic formula. If we can discuss this offline if you're interested. So going back to the practical, okay, so if you were bored about this part, or like if you got lost in the math, we can go back to discuss how this algorithm actually performs. And I have a few examples, and then I have the example of what happens in the marker selection problem. So in order to, this semi-definite program as I wrote it is not very useful in the sense that it's very strict. Like you have this strict constraint of this quadratic form being greater or equal than delta square for all points in the difference set. This is very likely to be unfeasible, or because asking that this is always satisfied may require you that you don't have any outliers, so that you choose this delta very small. So in order to be robust to outliers, one typical thing to do is to replace this hard constraint to a penalization in the objective and just penalize how many times you go below this threshold. And this is what we do here. Another thing that would be useful is to replace this minimization of trace to restrict to like go to a space of dimension at most R. So in the case of the marker selection, we know that we want 20 genetic markers, and in this case, we will restrict this trace to be smaller than 20. And so the number of constraints here is scaled with quadratically with the number of points. So in order to make this more scalable, something that we can do is we can sample the set of constraints smartly, like using k-nearest neighbors, or something that we do later is to use the centroids of the empirical clusters. And also, if you know that in what basis you want to select your markers, or like in what basis you want to find your projection. Like if you know that you want to project to a specific basis, then you can restrict this to a linear program. And that's what we do with the marker selection program. So this is just a proof of concept where I want to separate the 9s and the 4s of handwritten digits. And something that is interesting is that the projection that you find doesn't allow you to see like exactly what number it is, but it's enough for the classification purpose, and it actually performs quite well. And now, when going to the marker selection problem, we were interested in this data set where we had, here is a smaller data set where we have 4,000 gene expressions of 3,000 different cells. And the task is to find markers that determine the cell type. So finding the markers that determine the cell type is to look for a projection to a small dimension space where you know that the projection consists of selecting 20, say, of possible 4,000 genes. So you can think of it as a projection to 20 elements of the canonical basis. So the projection that you're looking for in marker selection is actually a diagonal, right? So you can think of it as a linear program. And you can say, now, M is going to be a diagonal matrix that is decided whether you pick gene I for I equals 1 to 3,000. And you're going to, instead of minimizing the trace of M, you're going to minimize the one norm of this diagonal vector of your matrix. And this is a simple linear programming formulation of the marker selection problem, but it's also a natural simplification of the SqueezeFit problem. And there is a very huge runtime difference between running a semi-definite program and running a linear program. That makes the problem to be actually feasible for these large single cell data sets. So we also do we do small modifications of the formulation of the linear program to introduce a slack variable to allow for outliers. So this beta is how much you went below this delta square threshold that you prescribed, and you minimize how much you went below that threshold for each point. And since we know that we have a target of number of genes to select, we can say that that the L1 norm of this alpha vector is going to be smaller or equal than S, for S, the number of target of genes. So you can put that as a constraint. And this is the direct formulation from SqueezeFit, just restricted to this this problem where we know what the basis where we want the projection is. But there are other ways to change this formulation. Instead of looking at all pairwise distances of points from different sets, which don't scale very well, we consider alternative implementations that aim for the separation of the empirical centers of the clusters after projection. This idea has been explored before, and there's this paper called NUMAX. This assumes that the classes are linearly separable in a way, which is not a bad assumption in these single cell data sets that are in such high dimensions. For the example of the concentric circles, that wouldn't work. But for these data sets, it actually works quite well. And it scales very well. We have a Python package that you can pip install that is called scgenefit. And if anyone wants to use it and have questions or bugs, I maintain this package for now. So yeah, it's actually very nice. And we can show for different examples that that it outperforms the state of the art. The state of the art in this problem usually consists on looking at this one versus all technique, where the idea is to identify one gene that one gene per class that has the most variability, meaning that if you look at the distribution of the gene in that class, and you look at the distribution of the gene in the rest of the classes, you want the gene that maximizes the distance between these two distributions. And so they identify one gene per class, but we observe that sometimes it's not that useful to identify one gene per class, but maybe a couple of genes will give you a much better understanding of the landscape. And we can see that when the accuracy of a classifier in a test set actually improves a lot when you allow for more markers than the number of classes. But still, the number of markers that we use is within the is within what is needed for this problem. So for instance, we can also extend this to hierarchical clustering, and also we can identify what I was saying before, the fact that maybe this gene is concentrated in these classes and not here, but then it complements with a different gene that is concentrated in a subset of the classes where this one is concentrated, to identify, let's say, this class. Like, you can use this as a dictionary in a way. So the final, we have like 10 minutes left, and the final thing that I wanted to discuss is the matching and alignment problem that is also related with this sort of data sets. Say that you have two compact matrix spaces or two data sets where you have pairwise distances. How how do you compute the distance between these two data sets? Usually what you would need to do is you would need to put these two data sets in the same space and measure the distance within the same space. So here, say, you consider all possible isometric embeddings to a common space Z, and then you compute the distance in Z, and you will look for the embedding, optimize for the embeddings that minimize the distance between f of x and g of y in this common space C. So, and the distance that you can use in C is the Hausdorff distance, or you can use optimal transport. So an example of this could be the Gromov-Hausdorff distance, like between metric spaces, which is the most common and useful distance between metric spaces. It has good properties, like the Gromov-Hausdorff distance between two metric spaces is 0 if and only if they're isometric, and things like that. Computing this Gromov-Hausdorff distance is simply hard, but you can formulate to as an integer program if your data sets are finite. And you can consider convex relaxations. I studied some convex relaxations of this problem before, but they are computationally very expensive. It's like impossible to run most of these convex relaxations in a reasonable amount of time for many points. Another example of this is the Gromov-Wasserstein distance, that the distance you consider in Z is based on optimal transport. And these algorithms are much faster. There are accelerations through this synchron algorithm, but they're still unfeasible if you are looking at a very large data sets in very large dimensions, like these single cell data sets that we are using. So what we propose is this approach called MREC, that basically what it does is just recursive matching. So basically, I don't know what matching algorithm I'm doing, but I'm going to use that as a black box. I'm just going to take my two data spaces. I'm going to cluster, consider K clusters in these two large data spaces, consider the centroids of those clusters, and then I'm going to match these K centroids using my favorite method for matching. And then I'm going to open up each of these clusters, and I'm going to do the same thing within each of these clusters. So if centroid 1 got matched to centroid 100, then I'm going to see all the points in that class and all the points in that class, and I'm going to recursively do the same with these subclasses, and then I'm going to find a matching in this recursive way. This method has theoretical guarantees provided on what guarantees has the black box matching algorithm that we have. But basically what you can say is that if your data is naturally clustered, then you can bound the distortion introduced by this procedure. But these theoretical guarantees are not very strong, and they can for sure be improved. But the nice thing of this recursive decomposition thing is that you can actually run it in very large data sets where sometimes the state-of-the-art procedures cannot be run because they're just too slow. And for different data sets, here I show for different data sets how does the accuracy improve when I increase the number of centroids. So for instance, some of these data sets are in the same space. So I just use the optimal transport distance as a black box in my recursive decomposition. But some of them, like in these two, these data sets come from different modalities. So they're not in the same space. So I have to use the Gromov-Wasserstein distance for that. And for instance, in this one, the accuracy doesn't increase at all. This dashed line is the baseline for the optimal transport without doing the recursive decomposition. And then we see that in order to approach this, we need to use like 3,000 centroids. So I think that there are some consistency results that could be proven for an algorithm like this, and theoretical guarantees that could be proven from analyzing clustering lower bounds. But we can actually run this in large data sets and obtaining a reasonable performance in a reasonable amount of time. So that's pretty cool. And the current extensions that I'm working on are combine these two ideas, this marker selection and this matching, to perform both things at the same time in an unsupervised way. So say you have two data sets, the single cell expression with the chromatin accessibility, and you want to find the subset of the chromatin that is the markers in the chromatin that would allow to do the best matching with the single cell. Or maybe you want to perform marker selection in both data sets with alignment at the same time. And also something else that we are working on is to extend this idea of marker selection, but instead of the marker selection be used to identify the clustering structure, we want to use it for other purposes and find it in an unsupervised way, for instance, for data reconstruction. And in order to do that, we need to use this selection in well, we use neural networks, so we need to see how we can think of the subset selection as neural network friendly constraints. And there is some sampling techniques that are differentiable that we are using for that. And other open problems that are related with the things that I just talked. So when is this projection factor recovery possible? Can we prove more instances of this theorem where we can do this projection factor recovery reconstruction? So the SqueezeFit algorithm in general is very inefficient because you have to solve semi-definite programs. So I could run it on MNIST, but I cannot run it in a large data set because this semi-definite program is not very efficient. However, there are non-convex approaches that one can use to accelerate these methods, or maybe one can exploit the structure of the semi-definite program to find another relaxation that is more efficient, and that's a possible direction of research. And of course, this consistency, like this is the performance guarantees for this recursive decomposition that I just talked about. So this is it. These are my references. Thank you so much for your time, and I can take questions if there are some.
Thank you very much for the fantastic talk. Exactly. So if people have questions, please put them to the chat, and then we will unmute you. You can also just put, I have a question. We'll unmute you. And there's already one question, which is, can we see the refs again?
Oh, yeah.
And before there are more, maybe I can ask one question to allow people time to type up. So I was wondering, so your problem, the basic problem is that of finding a good projection that kind of separates the data. And so in my interpretation, you could also see this as finding a projection that in a sense minimizes the variation of the data. So you have a large margin between two classes, and then in between there's nothing happening. And so in that interpretation, you could even apply this to a regression problem. Yes, so I was wondering, can you have a projection of just data points with labels, could be any number, not just 0 and 1, that kind of minimizes the variation of the data? Is there anything that you can think of that one can do there?
Yeah, I think that that naturally happens if the, I mean, with this SqueezeFit definition, if the data has some variation in a direction that does not that the other data sets are not separated by that in that direction, then this minimization would actually collapse that.
Okay.
So I don't know how it could be connected to regression, but
no, I'm saying I have points, and they're sort of labeled with real numbers. And now I want to find a projection so that they are nicely ordered and they also don't have that high of a variation. But you said that would essentially naturally happen.
I think so. Yeah, I think that would naturally happen if that direction does not is not relevant for separating two sets. Then that direction is going to be collapsed.
Right.
Mm-hmm. Yeah.
Okay, so I'm just going to unmute someone, if that's fine.
Mm-hmm. Of course.
I'm obviously not sure if Hans Johansson wants to speak.
Hi there. Thanks for doing that. I understand we have to keep the mutes under control. I just wanted to ask, for your linear program, you mentioned L1 norms to try to get, I'm assuming, sparsity. Have you looked at other norms like infinity norm or even fractional norms?
The infinity norm is hard to, like in the objective, is hard to, I think the constraints can be used, but the thing is that when you, in my experience, when you put an infinity norm, the algorithm is much slower because there is an issue with the differentiability there. Even though none of them are differentiable, I have the impression that is not, it won't work. But I haven't looked into it. The like weighted norms, yes, that's something that definitely could be useful. But I haven't looked into that yet. What we did look into was considering different possible set of constraints, and we explored that a lot, and we basically obtained that almost any constraints that you want to put, instead of just do pairwise constraints, that is very costly because you have too many. Just use constraints depending on, like say, fitting some sort of Gaussian into your data set, and use some constraints between the centers of these two different clusters, and also consider how big these classes are in order to have constraints from there, and those, all of them work quite well.
There is another question from the chat. Do we know how? Do we know which dimension will separate, or do we need to try them all?
So that's what we do, right? So what we're doing is we're gonna find them all, like we're gonna test them all at the same time by running this optimization technique. So if you wanted to just find an orthogonal projection, then that is hard because you will need to choose the bases and then kind of pick them all at the same time. But what we're doing is we're convexifying the problem and then exploring in this convex space that includes projections, but not only projections. And then you're doing kind of like an interior point, like you're moving inside this convex space, and you are continuously optimizing for all directions at the same time. Does that make sense? Does that answer the question?
I would say so, but if not, please.
Like you're looking at all possible combinations of these directions, and then you're moving continuously in that space, and you're finding the one that gives you the largest separation between your classes.
René has a question. So I unmuted you.
Yes, I was wondering in the formulation of SqueezeFit, how is, in many machine learning methods, there is a pre-processing of the data, normalization and whatnot. And that is, in this case, I would imagine that if the data is scaled, then I need to adjust the delta, but a priori I might not know the delta. So how is data scaling, pre-processing, and selection of delta handled?
Yes, that's a very good question. So in the practical applications that we have, what we do is we use cross-validation to choose the data, the delta. So what we do is, since this is a supervised problem, we just separate a subset of the data for test, and we do cross-validation for the choice of the delta that gives you the best performance in this holdout test set. But that's not, and we optimize, we use like some dual annealing for optimizing in that space, because since each evaluation of your function with a different delta is very costly, we just we explore the space with the limited number of samples of solutions of the problem. But that's what we do. But that is not necessarily the best way to do it. Maybe there are other ways we can do this that are better. Thanks, René, for your question. Does that answer it?
Sounds like it. There's another question by Zachary. Can you ask it?
Thanks for your talk. I was wondering if you've looked into making the set of constraints smaller by only considering some carefully chosen subset of the different set rather than considering the entire different set?
Yeah, we did many choices. One of the things that we did was for each class, we considered the nearest neighbors to each of the other class. That was like an implementation that we did that seemed to work well. But actually, we can show that, like mathematically show that when you write this semi-definite program, most of these constraints are not tight. The number of constraints that are tight scale with the dimension at most, but not with the number of points. Like if you have too many points in lower dimension. And so, yes, it makes sense to assume that actually you can do a smart sampling of the set of constraints and the solution would be exactly the same. The question is, how do you find those constraints? And we have some heuristics, but we don't have any proof on that.
Gotcha, thank you.
Thanks.
I have another practical question for me because I want to apply this eventually. So you said that in this linear program relaxation, SqueezeFit LP, it takes 2 seconds for 100 points in a 500 dimensional space. What if I had 1,000 points in a 5,000 dimensional space?
It also, I mean, it depends on what is the set of constraints. That's what makes the problem harder. If you have like n squared constraints, then that's not going to scale. But if you choose the constraints based on like the centers of the classes, then we can actually run. So in order to submit this paper for computational biology, we actually had to show that it scales to 10,000 points. 10,000 dimensions and 1,000 points, because that's kind of the scale that they wanted, and that I can run in my laptop in 10 minutes. So it's 10,000 dimensions, 1,000 points in 10 minutes in a laptop. I bet that you can do better.
Not on this old machine. Okay, so there's another question. I will just unmute. Everyone is now can now unmute themselves. So I just did that. So if you would like to ask the question yourself right now, please do so.
So there's a question here that I see in the chat by Roosevelt, that asks, how do you usually choose how many markers? So for instance, I mentioned 20 as an example, but what if you need more markers? And my answer to that, I think, would be from this plot. So you can actually see how does the performance of your classification on a test set changes when you increase the number of markers. So you can use kind of like an elbow rule to say, okay, when the marginal benefit of adding a marker is not that much, then you can say that you settle to that number of markers. It's kind of like the same idea that you ask when you say how many clusters should I be looking at. So, but a lot of times this number of markers limitation is given by the application that you focus on, which I'm not an expert on at all.
Makes sense. Thank you. Thank you for your question. I have one more question if there is time. Since the data is labeled, I was wondering if it makes sense or if it would be helpful to organize it into a tensor where each slice corresponds to one of those classes, and then maybe try to evaluate, like estimate the rank of that tensor, or maybe like try to find discriminative patterns in the data using that, and then compare it with these. How does that compare? Like, is it something that could be relevant at all, or?
I think that could be relevant. I think that the purpose of that would be slightly different, because if you, I don't know, I don't know, but my intuition is that if you analyze this separately, you can do some dimensionality reduction, that kind of like a PCA in each of these spaces, but then what you want is you still want to preserve the difference between two different classes, which would not be given by something, as you said, maybe. I don't know.
Sense.
Yeah.
Thank you.
Thanks.
Okay, so from my perspective, it looks like these are all the questions we have.